Hello, good morning. Welcome to biology. This is Mrs. Mann, and today we're going to be looking at how molecules get in and out of cells. So our topic is really cellular transport. So I just want to focus on some of the pieces that we tried to help you understand by doing our POGIL. Remember the process-oriented guided inquiry learning. So it was inquiry-based, right? You looked at four different models and we asked you a series of questions to get you to think about what was happening to those molecules as you were seeing things move one way or the other. And so, really, we wanted to highlight whether things needed energy, whether something needed a protein, what were some of the rules that determined how things would move. And we gave you some new vocabulary. So, today, the goal is really to take all of those pieces, and I made this note-taking guide for you to kind of get all the main ideas right in one place for you. So then that way you can study this to help you be prepared for the next practice quiz, and then also to be prepared for the unit test coming up. So with that, let's go ahead and get started and just make sure we really can understand what we need to know about cell transport. So the first thing here that I've included is a question. We're saying, well, what is a plasma membrane and why is it important? And you may wonder, why did I use the word plasma membrane instead of saying cell membrane? And I did that on purpose because the cell membrane is really the outside of every kind of cell. That's the part that's covering, it's protecting the cell from things going in and out of it. But that membrane actually also covers a lot of the organelles found inside of a cell as well. So you remember your mitochondria, there's chloroplasts, there's a nucleus inside of a cell. All of those are also surrounded by a membrane. And we call it a plasma membrane because plasma is referring to the fact that it can move. That phospholipid bilayer is not just a solid surface that never moves, it's flexible. It can move in order to make that cell fit into different spots. Okay, so let's go ahead and just write down what we know about the membrane. So the membrane is made up of phospholipids. And then I like to just remind myself of what that molecule looks like. So it has this little circular head on top, and then it has, sometimes we call them little tails. So these are our fatty acids that are coming down. They are connected by a glycerol, and then there's a phosphate on top. So sometimes we refer to the circle at the top as being the phosphate end of a phospholipid. Okay. And so a membrane is made up of these molecules called phospholipids that surround and protect both the cell, so protect the cell and organelles. All right. And then with that thought about that there is there's some protection going on here, is this idea that the membrane is selectively permeable. Okay. So permeable means that something can get across some kind of divider. All right. So selective, meaning there are certain things that can get across and certain things that can't. So some molecules can go across a plasma membrane. And remember, we're referring to both the cell membrane or the membrane of an organelle, and some cannot. All right. And then the next word we need to be aware of is this idea, this term is concentration gradient. All right. And so that sounds really tricky and complicated, but really what we're getting into is that if we had an enclosed space, and we had a bunch of molecules on one side of that space, and there were less molecules on the other side of the space. This sets up a concentration gradient, meaning that there are more molecules on one side than the other. Okay, if there is a concentration gradient, then we can follow the rules of diffusion. That is the natural order of things, is that they would follow these rules. So our concentration gradient is that there are more molecules on one side than the other. And then our rules that we follow, this is just how things work, is that first of all, molecules move randomly. So movement is random. Molecules are not alive. They cannot make decisions about where they are going. So a molecule isn't going to decide, hey, I'm a glucose. My cell needs glucose. I'm going to move right into the cell. That is not how molecules work. So do not expect that. Instead, a glucose molecule is simply always in motion. So it is going to flow from an area of high concentration to an area of low concentration. Okay, so movement, molecules flow from areas of high concentration to areas of low concentration. And this will keep happening until equilibrium is reached, until equilibrium is reached. And it's always good when you can use a little rhyming to help you remember something. So molecules flow from high to low. Molecules flow from high to low. That is the natural order. So without us doing anything weird to force things to go differently, this is this is the natural order. And so this means that molecules are following along the concentration gradient. They're moving from an area of high concentration to low concentration. All right, so how molecules move, they follow some different ways of transport. So one way we refer to transport is that we talk about whether something is passive or on the other side here, that something is active. As you can imagine, passive is like when you wait for other things to happen to you. It doesn't require any energy, it just happens. So the two types of passive transport are called simple diffusion and facilitated diffusion. So simple diffusion does not require a protein. I've got an image right here that just shows the arrow shows you the movement. The molecules, there's a bunch of them over here. There's not very many over here. So there is a concentration gradient. The molecules are going to literally flow through the in between the phospholipids to get from one side of the membrane to the other. It follows the rules of diffusion. Therefore, it does not require energy. The examples of molecules that use this method that we talk about in biology class is carbon dioxide, oxygen gas, and water. So these three molecules from the POGL you learned, these are small polar or non-polar molecules. And that has to do with the fact that they can go through, they're small enough to actually fit through the phospholipid bilayer. And then the other one that I just want to give an honorable mention to is sometimes you might hear the word osmosis. Osmosis is just a type of simple diffusion where the only molecule crossing the membrane is water. And so you can get kind of some interesting things happening here. It has to do with our saltwater example that we gave you at the beginning of the unit too. So you can start thinking about how that might impact whether things go into or out of cells. All right, then our second type of passive transport is called facilitated diffusion. Facilitate means to help with something. So in this case, the thing that is needed to help diffusion is a protein. So then I have two pictures here. Both are showing facilitated diffusion. They are following the rules of diffusion, meaning that molecules are flowing from an area of high concentration to low concentration. Therefore, it does not require energy. The types of molecules that have to use something like this are larger molecules like glucose, as well as ions, meaning something that is charged. So the reason for that is because in the case of glucose, it's too big to fit in between the phospholipids. The only way it's going to get through is to go through something that we call a channel. So I'm showing you two different types of passive transport. One of them is just a channel protein. So it has a special shape that allows a particular molecule to go through it. Okay, so it's like a little tunnel to get from one side to the other of a membrane. Okay. And then the other one was the one from the Pogel, where it actually changes shape. So this is still a protein. It still does not require energy. But in the case of glucose, when it gets through, there's actually another molecule involved. It's a hormone. It does not need any energy, but it's kind of like a little lock and key. So the hormone touches the protein, and the protein then is able to change its shape in order to let the glucose flow from high to low. So again, this wouldn't work if there was no glucose on the outside here. Okay. And then finally, we go to our active transport. So these ones require energy. So active transport, in this case here, it requires a protein. And sometimes we call these a protein pump because it requires energy because it's going against the concentration gradient. So you have molecules flowing from low concentration to high concentration. And in order to do that, basically you are forcing a molecule to go from opposite how it would normally move. So in this case, you can see on one side, you have a bunch of that molecule. On the other side, you don't have very many. So if we're going against the concentration gradient, this molecule has to get pushed through, which requires energy. Okay, so energy is going to force that molecule into an area that already has a bunch of those molecules. So examples of things that might do this is large molecules and ions, but really it can work on anything that needs to be pushed against the concentration gradient. And then the last one I just threw in here because I think it's interesting. And I love this visual of endocytosis and exocytosis. So endo means bringing in, exo means bringing out, like an exit. But what's kind of cool is that the membrane itself can actually like change its shape and surround a bunch of molecules. So in endocytosis, it's trying to grab a bunch of molecules and pull it directly into the cell. And so it surrounds it with its own little plasma membrane. So we would call this a little vesicle or and so then now these molecules are inside of a vesicle inside the cell. And then also the opposite could happen as well, is that you could then have some of these, like you could have a vesicle that then attaches to the phospholipid bilayer, which then opens it up and releases a bunch of molecules into the outside, which is kind of cool. So anyway, hope you enjoyed this little lesson on cellular transport. Hopefully you feel ready for the next practice quiz. Hopefully you can talk about whether or not proteins are involved. You can talk about whether molecules are following the rules of diffusion, that you understand what a concentration gradient is. Test yourself, quiz yourself, be ready for that next practice quiz. Good luck and see you later.